You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Anthony St.Leger. He's an assistant professor, Department of Ophthalmology at University of Pittsburgh. So, Anthony, thanks for coming. How are you doing?
Good. Thanks for having me.
Yeah, it looks like you're working on the Ocular Microbiome, which sounds super interesting. I guess that's the microbial attachment to our eyes, right?
Correct.
Okay. Do most people even know that we have a microbiome of the eye?
So most people don't. I think whenever you talk about the microbiome, most people think about the intestine because there's been such a push on trying to normalize the intestinal microbiome to have the good bacteria be pretty prominent and maintain health and sort of prevent infectious disease as well as prevent autoimmunity. But about 3 years ago, we found using a mouse model of eye disease that there are actually bacteria that live underneath the eyelid on the eye, and they do similar things to what the intestinal microbiome does in modulating susceptibility to infectious disease and possibly autoimmunity.
Yeah, I wonder if I don't know if this has happened to you. For sometimes I've seen like I guess they call them floaties, or if the light is right, I feel like I'm seeing things maybe on the surface of my eye, but in my field of vision for a moment, I see things like floating by. It happens every once in a while. I don't know if that's me seeing any of the microbes in my eye or what that is.
So that's probably just proteins inside your eye that are floating in front of the retina. Is what I would think. Since the bacteria are so small, it's probably pretty unlikely that you would see them actually across your field of vision.
Oh, okay. So in the eye, how many distinct niches are there? Are there ones that always stay beneath the eyelid, and ones that are on top of the eyelid and the center of the eye?
So what we know around the eyelid is skin. So it's actually more like the skin microbiome. So there's a pretty consistent diversity of bacteria that live on the skin. However, when you get past the eyelid and get underneath the eyelid, the diversity kind of constricts. And we're just beginning to understand what controls that contraction of the microbial species, and it looks to be affected by your geographical location, so where you live, whether you wear contact lenses or not, your age, it changes, as you age, and whether you have any sort of ocular disease, like if you were to have dry eye symptoms, that appears to affect the diversity of bacteria at the eye. But we're really not sure why it changes from the skin to the eye. I think that's one of my major goals in the lab, is to try to figure out what allows bacteria to stay underneath the eyelid, and what factors allow it to thrive there, and what benefits those bacteria produce for the eye.
Well, I guess the eye is, we blink, I don't know how many times a day, but that kind of like windshield wipers or bathes our eye surface in various fluids, and do the bacteria feed off proteins or things like that, the nutrients that are in that fluid, or like what's some of the dynamic you figured out?
So what we've seen is, and what's sort of been conventional thought in the eye field, is that the tears actually are very harsh on bacteria. They contain a lot of enzymes and factors that kill bacteria on contact. So your eye is actually very well protected against bacteria landing on the eye and infecting. And then as you imagine tears wash across the eye, that also helps to wash away the bacteria. So the eye is actually a very inhospitable place for bacteria. And it's sort of illustrated that if you were to take a swab of your central cornea, so the middle of the eye, that routinely comes back devoid of any bacteria because the cornea is a very not good place for bacteria. But as you go underneath the eyelid, the flow of tears is a little less, and you're less exposed to the environment. It looks to be a little more gentle, so the bacteria have a chance to adhere to the tissue there and grow and to survive and almost thrive. It looks like that something in the tears actually helps them to live on the eye, and we're trying to figure out what factor might be causing that.
Well, if you're actively tearing or crying, is that very different from the normal, little micro squirts of tears that are continuously coming out of the ducts in your eye?
Yeah. So if you're actually crying, the makeup of those tears are slightly different than just the normal steady state tear. So I've tried to make people in my lab cry with my terrible jokes, but we don't sort of see the same effects.
So dad joke tears are different from tears of despair.
Yeah, right, exactly.
Interesting. I don't know if you can profile this, but are the bacteria different that are right in front of or on top of the pupil area versus the iris area versus the sclera?
So we have only been able to find it underneath the eyelid. There really doesn't appear to be any sort of bacteria adhering to the eye in front of the pupil or in front of the iris. We've really only seen it underneath that eyelid, and I think that's where most of any adherent bacteria live. And we've, from our studies, we've only been able to find one type of bacteria that can colonize the eye. I'm sure there are others, and I'm sure people are looking at it, but the only real bacteria that we've seen that's able to adhere to the eye and stay there long term is part of the genus Corynebacterium, and it sort of grows in a filamentous pattern underneath the eyelid.
So there's no bacteria on when your eyes are open. There's no bacteria sitting in the liquid that bathes your eyeball. They're just under the eyelid.
It could be introduced there if you rub your eye, or if it flies in there from the environment. It could be there, but there's been extensive studies looking for bacterial adherence and bacterial DNA in the center part of the eye, and it always comes back as negative unless you have some sort of really severe ocular infection.
So maybe blinking is like the street sweeping that takes any materials that land on the eye surface and pulls them under the lid so that bacteria can work on them.
Right, exactly. That's certainly possible. And I think, as you age, that definitely plays a role in how you blink and how often you blink. Because an interesting feature we found with our bacteria too is that we had to give it to animals very soon after birth, because as they age, the eye becomes actually less hospitable. So you lose the ability to colonize the eye with bacteria as these animals age. So we're trying to figure out that dynamic too. What's the optimal time if you were going to give some good bacteria to people or to animals, when during childhood or adolescence or adulthood is the best time to actually give them those bugs.
So the bacteria that live under our eyelids, like what kind of metabolites do they make, or what have you been able to glean about what they do?
So we've done some basic characterizations, and that's sort of a goal of the lab right now, is to try to figure out what they actually do. And right now we're not quite sure, but they do metabolize things in the tears that they use as energy sources, and the immune system actually recognizes them, and that recognition actually boosts the protective ability of the eye. So basically, when the bacteria are there and the immune system recognizes it, that the eye is less susceptible to be infected with other more serious pathogens that cause blindness, like Pseudomonas aeruginosa can destroy the cornea very quickly, or Candida albicans can infect the cornea and cause a lot of disease. But what we see is when the bacteria are there, something they do produce that's recognized by the immune system helps protect the eye. So indirectly, something they produce protects the eye from infection.
people that wear contacts, I would guess that produces a different microbiome. Have you studied that? What's different about people that wear contacts?
Yeah. So my lab has not studied it, but others have, and what they see is the makeup of the bacteria of the ocular microbiome is totally different, and there's actually more pathogenic bacteria in those people that wear contact lenses. So it definitely shifts the microbial landscape so that different bacteria are represented. And I think there's studies now trying to figure out how you can sort of normalize the bacteria so that you don't have such a dramatic effect.
Yeah, I've heard that people that watch screens, their blink rate slows down, and that can cause the tears and all that in the eye to dry out because they're not blinking enough to lubricate the eye. Anything you've heard about in that regard?
So in regards to the microbiome, we haven't heard much, just because looking at the functional effects of the microbiome is so relatively new. But we do see that whenever we induce sort of a dry eye phenotype in animals, that the ability for bacteria to colonize is also shifted. So the effects of a dry eye do change how bacteria can live at the eye and what they're actually doing, whether they're beneficial or could they turn into sort of a pathogenic type of bacteria.
I don't know if there'd be any effect, but people that wear glasses versus don't wear glasses, do you think that would affect their microbiome of the eye, or is that too subtle to have any change?
I don't know about that. It's possible. I don't see how it can change too terribly much, just because there's nothing really changed about the eye. But I guess anything is possible with, whenever you're affecting vision, it could affect the microbial landscape.
Yeah. So what experiments are you doing right now that you think are going to give you some good data? What do you feel like you're on the edge of figuring out?
So we're currently doing experiments right now to see, what these bacteria eat. And it looks like they eat something in tears that allows them to thrive. I think that sort of gets us into an idea of what factors allow good bacteria to stay and helps to keep bad bacteria out. We've also been moving towards making our system more physiological. We're always trying to move towards helping humans. I think that's the goal of every scientist, is to help humans. And so the original bacteria we have was isolated from mice, and we showed a lot of beneficial effects of this bacteria in mice, but we never really knew if this bacteria or a similar bacteria exists in humans. So we have a UPMC eye center here that it's basically our eye clinic, and they have a humongous library of bacteria that's been stored over decades of patients. And Regis Kowalski, the director of this, provided us with over 30 clinical isolates of bacteria, and we've been able to screen through those isolates and identify which human isolates can be colonizers of the eye and how similar they are to the mouse isolate we have. And interestingly, we found a handful that are very similar to the mouse isolate we have, suggesting that the effects we see in mice also apply to humans. So now we can take those clinical isolates and possibly modify them to become almost like probiotics to alleviate dry eye disease or other types of diseases of the ocular surface.
If someone has contacts, instead of just giving them, I don't know what they give, saline or some kind of solution, if you had a probiotic part to it, that, if you knew what healthy eyes had and you added that into the drops in their eye, it might help.
Right, yeah, exactly. And interesting about eye drops too is that what we find is the good bacteria actually metabolize those eye drops too. So there's something in the eye drop as well as in tears that helps to create sort of foster a good environment of bacteria.
Cataracts, what have you studied those, or have other labs? And is a cataract, is there a biofilm component, or what's interesting about them?
So in the causes of cataracts, we really don't know if there's any sort of link, but I think whenever you cut the eye open to alleviate cataracts, I think you're always concerned about whether you're introducing bacteria that should be staying on the outside of the eye, inside of the eye. So we know that the good bacteria, whenever it's on the exterior part of the eye, is good. We don't know if what happens if you put a needle or cut the eye open and accidentally get some of those bacteria inside the eye, then what would happen? We haven't done those experiments, but I think that's at least relevant to human disease, where you're having, countless cataract surgeries a day, and trying to, mitigate any sort of bacterial infection.
And I don't know if mice ever have cataracts or they could be induced, but it might be interesting because maybe they have their own microbiome or something interesting about them,
Right. So usually we only see cataracts form after they die. So once we sacrifice them and perform experiments on them, they will get cataracts. But we never, they don't routinely happen in the live mice.
Huh.
Yeah.
How hard is it going to be to figure out what the bacteria are eating in tears, and how would you figure out that dynamic?
So I think the goal is to try to find a bigger surplus of tears. We just need to be able to have enough of a supply, then we can split it up into different, it's called fractionation, and you can figure out exactly what part of the tear is used to be metabolized. It just takes a little bit of time and a bigger supply of tears. So that's what we're trying to go after right now.
So you're using mouse tears?
Yeah. So we use we can either isolate mouse tears from washing the eye, or we take their lacrimal gland. So it's the main tear-producing gland that's just outside the eye, as sort of a surrogate. So all of the factors that would be produced that come out in tears are also in the lacrimal gland. So we use that as a surrogate to sort of do some of our initial screening.
Very interesting. Any other projects or research that's going on that you're looking at, eagerly waiting for it to be completed, that would inform your own?
So I think another sort of unappreciated aspect of the eye is that it's the most densely innervated tissue of the body. So if you think about, whenever you get a hair that lands on your eye, how disruptive it is and how painful it could be, that sort of illustrates how many sensory nerves are in the cornea. What we're seeing now in some of our infectious disease models is that things like viral infections actually cause those sensory nerves to retract. And that's a relatively, significant issue in humans, because if you lose the ability to blink, then you're more susceptible to dry eye, you're more susceptible to traumatic injury. And it's possible that some of these, the increase in dry eye symptoms that you see all the time that are primarily attributed to looking at a screen is, could be that you're losing a lot of these sensory nerve endings in the cornea, presumably due to screen watching or other things that are causing some issues in the ocular surface. So that's what we're trying. That's another arm of our research, is to try to see how the neuroimmune axis is functioning at the eye to preserve vision and just preserve quality of life.
I wonder again if anyone's looking at, I don't know what the tear composition is or what the microbial composition is for people with glaucoma or cataracts or after eye surgery, or there's I guess a lot of different things that can go on with the eye that can maybe inform you what happens normally. Pink eye, that kind of stuff. Yeah,
so pink eye specifically is interesting because with conjunctivitis, I think we all know how substantial the issue is with conjunctivitis. If anybody has any kids, how life-altering it could be whenever you have a kid with pink eye, but you take them to the doctor and the doctor automatically prescribes antibiotics. And a recent study showed that 60% of pink eye cases have an antibiotic prescription. However, a large majority of pink eye is caused by a viral infection. So it's self-limiting and really doesn't have any treatment. So basically it basically contributes to this overprescription of antibiotics. And we really don't know how those antibiotics are affecting the microbiome and how they're affecting ocular health. And I'm not advocating for not using antibiotics, but I'm just saying that we might need a more judicious assessment of whether pink eye is truly caused by a bacteria or it's caused by a viral infection. So we can prescribe antibiotics a little more, I would say, conservatively.
Are there any fungi or yeasts or phages that have been observed in the eye?
So we've seen there's been infections with Candida albicans and other sort of fungal species. Aspergillus is a fungal species that infects the eye. really looking at the fungal, it's called a microbiome, hasn't really been done at the eye. It's been done for the skin and other tissues, but it hasn't reached the eye yet. There is a lab that has looked at the possible viral component of the microbiome, and what they see is it's relatively conserved across humans. It's called torquetenovirus, and it's linked to some cancers, and I think we're now just trying to figure out, why it can be propagated in the eye and why it stays there long term. But I think that sort of illustrates that, yes, there are viruses that are routinely found at the eye, as well as fungi. We just need to actually look. I think we just haven't looked yet because our focus has been on bacteria for the time being.
Also, what about the lower eyelid versus the upper one? It doesn't seem like the lower one really moves. The upper one moves a lot. Are the bacteria different there?
I don't know if the robust studies have been done on whether the upper eyelid is dramatically different from the lower eyelid. From what I know,
it's like northern South Korea. There's no man's land with the
eyeball
in between.
Demilitarized zone. Yeah, I think it probably ends up normalizing at some point, because if you think about when you fall asleep and your eyes are closed for a long period of time, that the bacteria probably eventually sort of get distributed throughout the tissue.
Yeah, I just wonder if they have to be different because one's moving a lot, one's not.
Right. Yeah, it's possible. Yeah.
Yeah. Okay. Well, very good. So, what's some of the near-term goals you have for your own research?
So I think some of our near-term goals is to start sort of disseminating what we've been finding in the uniqueness of the bacteria and the uniqueness of the immune response at the surface of the eye. We've been looking at the makeup of the immune system at the eye of humans, and we've been finding some interesting dynamics whenever there's different types of infections, like viral infections or bacterial infections, how dynamic the immune response is in the human eye. We've also been focusing on the bacteria themselves. That can we modify these bacteria to act as drug delivery vehicles? Can we modify them to produce factors that possibly act as a lubricant for the eye to better treat dry eye disease and things like that? So we're trying to do some innovative thinking that we can engineer these bacteria to be, definite probiotics instead of just regular bacteria.
Okay. Well, very good. Well, Anthony, any other questions that you wanted me to ask? And if not, where can people find out more about your lab and your work? So we have a website.
I don't really have it right now. I can send that to you later. But we have a website on the UPMC Eye Center website that describes sort of our lab interests, what sort of other projects we have, and we're always hiring. So any scientist of any level we're always interested in getting some great talent working on our stuff.
Well, very good. Well, thank you for coming. I appreciate it.
Perfect. I'm glad to be here, and I appreciate you asking me to be on the show.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.